So I'm very pleased to be here. I think that what you're going to hear is a harbinger of what will come. And it wasn't mentioned earlier, but I think that we owe Ralph a great debt for getting the whole process off the ground. I mean, it's amazing from a little seed corn what the mighty oak has grown. Not only that, but he's also the founding director of the Center for Brain Activity Mapping here at UCSD, and he's already told you about some of our activities. But he's also the director of CalBrain and now the local alliance, and so he's a one-man organizer.
He also vacuums.
Okay, well, this is the way a neuroscientist looks at the brain. It's not one problem, it's literally thousands and thousands of problems wrapped up into this enigma that produces all of our thoughts, hopes, and the great things that have come down through history that the brains have created, human brains, science, technology, art, all the wonderful things in life, culture. And it really, the reason why it's such a difficult problem is multi-scale. This is the level of molecules, and this is the level of the entire brain, central nervous system. There's 10 orders of magnitude, spatial scale, right? That means that you have to fill in every single level here with some way to make measurements and understand how many, there's 100 billion neurons, there's 1 million billion synapses. This is a real challenge. It is indeed the last frontier. And Ralph very nicely pointed out that when you have a device that has so many moving parts, you're in trouble if you want to repair it. So I remember when I was young and my father let me take apart a watch that wasn't working. And that was the easy part. Because that's what neuroscientists are good at. They're good at taking it, finding out all the molecules and parts lists and so forth. But putting them back together is really going to be the challenge, to try to understand how all those pieces fit together. And when one of the parts goes wrong, how does it percolate up and to produce the deficits that we see here in mental disorders? That's the challenge. Now, as Ralph pointed out, when the Brain Initiative was announced a couple of years ago, what was interesting, I think, and this is part of the reason why it was actually chosen over a lot of the other potential grand challenges, was that the goal is really innovative neurotechnologies. You notice here, the goal is not to cure Alzheimer's. The goal is not to help directly help people with depression. Every year, NIH spends $5.5 billion on those projects, and we're not making progress fast enough. That's the problem. The problem is that it's not working. The drugs we have are inadequate, and the techniques we have are really backwards. They're the ones that we can use on humans. So the key here was innovative neurotechnologies. And as Ralph said, that means we need the help of the engineers to help us develop new techniques that will accelerate the research so that instead of taking 100 years to be able to come up with some new approach to depression, it only take 10 years. That's the goal, to develop those techniques. That will make it possible not just for mental disorders, but for understanding how the brain works and who we are. And this is going to be a fantastic moment in human history. It's like landing on the moon for the first time, understanding the basics of how we think. Now, the one technology I want to mention in my talk was one that was, Ralph made a long list of them, and there's a lot of different things you can do. But one of the biggest advances in the last decade has been what's called optogenetics. Now, neurons have electrical signals, and the way that traditionally you record is by putting a metal electrode down and touching the tip to the neuron and eavesdropping on its electrical signals. Well, it's now possible with optogenetics to target specific neurons with genes, in a particular gene called channel rhodopsin, which when you stimulate it with light will produce an electrical signal. So this is really remarkable because it means that we have direct access to particular cell types in the brain with light. Now this is all being done, as you can see right now, with rodents. But in the not too distant future, there will be ways of being able to target specific neurons in human brains. And that would be light fantastic, to be able to not just to be able to go in and selectively manipulate the neurons that we know are probably causing a disease like depression, but to be able to right it and rebalance it and get the brain back on track, similar to deep brain stimulation, but using light instead of electricity and much more selectively. Okay, I want to just to show you, I mean, this is just this is actually a very important discovery that was made at Caltech by David Anderson, but I just want to show you the power of this technology. So this is, what you're going to see is a mouse that has had a channel rhodopsin put into specific neurons in the hypothalamus. The hypothalamus is part of the brain that regulates things like food and sex, survival, fighting and fleeing, and so forth. And what he does is when he turns the blue light on, he can change the behavior of the mouse, because this is a part of you'll see, this is a part of the hypothalamus that controls the things like fighting and mating. So here's this is an unreceptive female. The light goes on, and you can see that the mouse is not taking no for an answer. Interestingly, there's another movie I'm not going to show you, but it turns out that even if there's no female mouse, but there's just a balloon, this male mouse will go after the balloon. Clearly very motivated. Okay, now this is exactly the same mouse, but now what they did is they turned up the light intensity. So this is the same neurons, but there are more of them now that are firing with more electrical activity. And let's see how different the behavior is. Did I get the right one? No. Let's try. Okay, let's. Okay, so here's this nice little lady mouse, and now the light goes on. Exactly the same part of the hypothalamus. The only difference is that the more neurons are firing, and you can see this little mouse goes ape. I mean, it's really sad. So it means, this is amazing, that the very same part of the brain, the very same neurons, can switch back and forth between mating and fighting. That's a mystery. How could that be? So that just shows, I mean, it's an important discovery. We're just beginning to appreciate the power of the selective ability to go into the brain and stimulate a particular neuron. Now, right now, it's working extremely well in rodents. We're just beginning to translate into monkey brains, and John Reynolds at the Salk Institute is one of the leaders in that area. But eventually, what we like to see is this technology translated into the operating room. Right now, the technology that's used, you saw the deep brain stimulation, those electrodes were approved by the FDA 30 years ago. We have so much better techniques that are being used in rodents, but it's hard to bring them into the operating room because there's a big barrier. It's as expensive to develop a new device as it is a drug, which is hundreds of millions of dollars. And so there's been a reluctance to do that. But that's all changed now. The FDA is now part of the Brain Initiative, and they are going to reduce the time it takes to get an experimental permission to go in and put a device like the electrode array into a human brain. From now, it takes years, but they're going to take that down to 3 months. So that's going to greatly accelerate. Even if we just take what we now have and put it into the operating room, it's going to be a renaissance in terms of what we can do. So this is the future. Now, the key to the Brain Initiative, as I said, is to accelerate progress And you can do that by scaling up. That means doing what's now done in individual labs, one neuron at a time or one brain area at a time. And just like the Genome Project, create a whole array so we can now interrogate all the genes at the same time. We need to be able to access every cell type. We don't know how many neurons there are. We know there are, for example, just in the retina, 100 different types of neurons. We don't know how many there are in the cortex. The best guess is about 1,000. We need to be able to selectively stimulate or silence each of those types if we want to make progress. We need to, as Ralph said, scale up recordings. We can't continue doing 1 or 2 neurons at a time when there's 100 billion. We need to be able to record from 1,000, from 1 million, and that's what we hope to be in 10 years. And finally, without anatomy, without knowing how the neurons are connected, you can't make any progress at all, because that's the bedrock of the brain, is how it's connected. And so new techniques are being developed to be able to take the electron microscope, very high resolution pictures, serial section, and then reconstruct every single neuron, every single connection, and create a wiring diagram. Now, none of these by themselves is going to solve the problem, but if you do all 3 at the same time on the same brain, you can make progress. Now I'm going to give you one example from my lab just to show you what is possible now. And this is a collaboration with Sid Cash at Mass General. Sid Cash is a neurologist. He works with neurosurgeons. And what they do is they implant electrodes in epilepsy patients, hundreds of surface electrodes, that allow them to record and isolate the focus of where the seizure is starting. And that means they have to keep track and record continuously for several weeks. And this is something now, data that we have access to. Now, one of my interests is in sleep. So how many people here sleep every night? Okay, is anybody here who doesn't? Okay, one person. Okay, I want to talk to you later. We have some experiments to do. But no, it's really remarkable. We sleep 8 hours a night, every night. It's 1/3 of our life, and we don't know why. Lots of guesses, hypotheses, but the fact is that sleep is a mystery. Here's what we do know. We know that when you fall asleep, you go from a conscious awareness to a state of what's called deep, slow-wave sleep, and you can stay there for an hour initially. But during the night, you cycle between slow-wave sleep and REM, what's called REM sleep, rapid eye movement sleep. That's when you dream. And you go back and forth, and later in the night, you have more time dreaming, less time in slow-wave sleep. And the brain is constantly active. It doesn't stop. It's just going at a very different pace. And the activity patterns are completely different from the awake patterns. And there's one in particular. There's one pattern that occurs in this intermediate stage, stage 2. They're called sleep spindles. And they're very, very interesting because they're little packets, 10 hertz. They last for about 2 seconds. And they're one of the mysteries, and one of the hypotheses is that they're very important for consolidating memories, taking experiences you've had during the day and integrating them with all of the knowledge that you have and all of your previous experience, your knowledge about the world. And if you don't sleep and you don't get that integrated, they could have dire consequences. Like, your cognitive abilities degrade with sleep deprivation, and ultimately, if you don't get enough sleep for a long enough period, then you have psychosis. So this is, for a human brain, this is something that really is detrimental. But I'm going to focus here on these sleep spindles. And here's a sleep spindle. You can see that little burst of 10 hertz. And what you're going to see is what the spindle looks like on 64 electrodes being recorded from a human brain. So let me first explain what you're looking at. So this is the human brain seen from the side. And each of these little circles is an electrode. And the gray level of each of the electrodes tells you what the electrical potential was at that location. And this red one here corresponds to this little red dot, and you can see it's going up and down, and with this time scale, it takes this is very fast. This is a third of a second. So this goes by like that. But what you're going to see, though, is a pattern of dark and light. And here it's light and here it's dark, not just for this one electrode, but for all of them at the same time. And you're going to take a look and see, tell me what you see in this pattern, right? You're going to have to really pay attention to all of the different locations at the same time. Suddenly a lot of black. Isn't it interesting? The black seems to be moving. Everybody see that? Okay, this is amazing. I've been working on sleep spindles for 20 years, and in collaboration with people who study cats and rats. And this is, to be able to do this in human and see this pattern that's never been seen before in any species is amazing. It's telling us that there is a traveling wave every cycle of the spindle, and there are about 20 of them. You get this traveling wave that circulates around the entire cortex, like this. We call them Princess Leia waves. What does it mean? We don't know, but we're thinking that this may have something to do with what's called synaptic plasticity, that you can trigger synaptic plasticity by having inputs and outputs with a slight time delay. And as the thing rotates, there's time delays from one area to the next. So this could be a way to cause changes to occur in the cortex that are going to be permanent, that are going to reorganize the knowledge in all of these cortical areas. And this is a way of globally organizing all the areas so that they're all working together. Now that's a speculation, but the point is, without these tools and techniques, without being able to study humans, we wouldn't make as much progress as we are today. Here's another technique that really, within the last 10 years, has revolutionized our knowledge of what's called macroconnectomics. And this is the long-range connections. See, here's the cortex out here. And here is each of these little noodles is a fiber bundle of axons that goes from one location to another location, right? These are the long-range superhighways within the brain. See this red one here, right? It goes across the corpus callosum, and then it comes back up here. That connects these two areas, even though they're as you can see, this is a long-distance highway. It's really a dramatic, and then these white matter, they're called white matter because it's the myelin that makes it look white to the naked eye. But now with the very same MR, magnetic resonance, you do what's called diffusion tensor tractography, and in individual brains you can figure out how they're connected internally. And it turns out that they're different. Everybody has slightly different pathways. Not only that, but we now know that when you learn something new, the volume of these pathways changes. And we don't know why or how, but it's really remarkable. We've always thought the changes that were important were within the synapses in the cortex, the thin layers, a few millimeters on the surface of the brain. But now we're beginning to think that these changes within the white matter may be very important for timing, for the time delays of the signals between the areas. And that may be important for being able to organize the whole brain so that you can recognize a friend within 100 milliseconds just by previous experience and all the different parts of the brain integrated together, electrical signals integrated together. And I want to show you a movie. Now this movie is constructed to be as accurate as possible, but it's an animation of the kind you're going to get out of Pixar. But it really does, it's science fiction, but this is the future. This is what we're going to be able to do. We're going to be able to go into your brain and figure out what signals are going back and forth and be able to manipulate them and help restore function in cases where it's not working properly. And it's based on the tractography I showed you earlier, and it's based on electrical recordings, and so it's a combination of electrical and anatomical guesswork. So here's the brain, and what you're seeing, every little blip here is an electrical signal, the little light flash. And every time you see a large area light up, that means that it's been activated. Maybe this person is seeing something in the back of the brain, the visual system gets activated. And these long-range pathways that you're seeing are signaling like from the visual system to the prefrontal cortex where you're planning. You recognize somebody, but now you have to decide what to say to them, and then that part of the brain now reports to the motor system. The motor system is going to figure out what the words are that you need to say. So isn't it amazing what goes on in your brain? Or what doesn't go on sometimes. It's one of the advantages of doing a movie like this, is that you can actually dive down into the actual working part. It's also very beautiful, I mean, just to see the complexity. Only a very small part of it. So I'm going to end by saying that this picture was taken just minutes before the announcement. And I just want to say that this is a project that has now spread over the entire world. Every major nation in the world, Europe, China, Japan, and Korea, have announced some form of brain initiative, some form of what part of the task, because it's going to require everybody pulling together. And so everybody wants to do some part of it. And within the US, it turns out that there are a number of different agencies that are contributing, and there are more joining us every day. But I just want to point at a few of the people here. So first of all, you've heard about the Kavli Foundation. The Kavli Foundation got things off the ground. Well, that's Mi-Young Chun. She's the spark plug who actually brought the nanoscientists and the neuroscientists together and brought the white paper that Ralph mentioned to the White House, and continues to be a spark plug. Bob Kahn, former dean of engineering at UCSD, now the president of the Kavli Foundation. And very important, we have a Kavli Center here. It's called KIBM, Kavli Institute for Brain and Mind, and Nick Spitzer, who's in the audience. He's right there. You know, it's a little, when you have a laser pointed to your chest, you know, you have to worry what's coming next. And so the representatives here from the Howard Hughes Medical Institute. This is Francis Collins of the NIH, which is the anchor agency. This is NSF. This is Amy Gutman, president of Penn, who's the head of the bioethics committee. Really, really important that when we start doing experiments with humans, it has to be done completely above board. We have to be very careful what we put in brains. This is really critical. And the importance of it is illustrated by the fact that Amy is standing right next to the president. Okay, well, this might be a good place to end. I want to thank everybody.